My name is Guri Johal, and I'm a professor at Purdue University, which is in Indiana in the US. And I'm in the Department of Botany and Plant Pathology. By training, I am a geneticist as well as a plant pathologist. And my seminar yesterday was on a new technique that we have developed in maize, which we call corn in the US. And this technique allows us to induce mutations in corn at a very high level. What I mean by high level is that with our traditional approach, which was based on pollen mutagenesis, our new approach takes advantage of both pollen mutagenesis and seed mutagenesis and introduces mutations at a very high density in the genome of maize. And by inducing these mutations at high density, then we can facilitate certain genetic strategies which were unavailable to us before that. And with this approach, we can also create new varieties, we can create new traits, but we can also take some old lines, and if they have any flaws in any of their traits, we can fix those very rapidly within 2 to 3 years. And what that allows us to do is to introduce a lot of new variation in these allele lines without having to break down the allelic combinations that breeders have put together in them. So we have called this technique the next generation of mutagenesis, NextGem in short. This technique works really nicely, perfectly in corn, but can also be extended to other plant species which are not open pollinated, unlike corn. Many of the plant species, crop species that we work with, they are self-pollinated, whereas maize is open pollinated. But even with self-pollinated species, we can use this approach, which is nothing other than a recurrent mutagenesis approach. So instead of mutagenizing plants for only one generation, we can do mutagenesis over multiple generations, 2 or even more. And at every stage of mutagenesis, we introduce new mutations and more mutations, and that allows us to enhance the density of mutations. And when you enhance the density of mutations in your organism or the genome of your organism, then it will allow you to handle populations which are much smaller compared to the traditional level of populations. And this also makes feasible some of the screening strategies or some of the assays that you need to conduct to identify genes of interest much more effectively than before. So that's why we call it next generation mutagenesis, because it hasn't been done like that before, and it is much more effective now compared to before.